Good morning to all. In this video, I am talking about sexual reproduction of angiosperm plant. We know that flower is the reproductive structure of an angiospermic plant. So, the sexual reproduction of angiospermic plant can be divided into three main events: pre-fertilization event, fertilization, and post-fertilization events. So here I am only discussing about pre-fertilization events. We know that in a complete flower, there are four floral cycles. First two, that means calyx and corolla, are only the accessory cycle or helping cycle. The other two, androecium and gynoecium, are the reproductive cycle. So androecium is the male reproductive cycle and gynoecium is the female reproductive cycle. The single member of androecium is known as stamen, and it has two parts: the long cylinder filament, and at the tip swollen part is known as the anther. So within this anther, microspore are developed. The development of microspore is known as a microsporogenesis, and after that, from this microspore, male gametes are developed, and this process is known as a microgametogenesis. Like that, in female floral cycle, the gynoecium, the single member of gynoecium, we used to call them as a carpel or pistil. So pistil also have three parts. The basic swollen portion is known as the ovary. The middle cylinder portion is known as a style, and the tip portion, which has the receptive function, is known as a stigma. So stigma, actually it receives the pollen grain. So development of megaspore are within this ovary, and this is known as a megasporogenesis. And within this ovary also, the development of egg cells are happen actually in ovule. This process is known as a megagametogenesis. So when you cut the anther, in a single anther we found four microsporangium. Here we can see, when we transfer section this anther part, we found that four sac-like structure. This sac-like structure is known as a microsporangium. So when we observe this microsporangium, we can categorize them in a wall layer and this central middle portion as a sporogenous tissue. So these microsporangiums, what they have four type of wall layer: the outermost single layer, continuous epidermis, following that endothecium, generally 2 to 3 layer, after that middle layer 3 to 4 layer, and at the innermost single layer continuous is known as a tapetum. So tapetum has the actually nutritive function. They nourish the developing pollen grain. Formation of microspore from pollen mother cells is known as the microsporogenesis. Here you can see, when from anther to sporogenous tissue, that means when we transfer section the anther, we found four sporangium, and the central portion of the sporangium we used to call sporogenous tissue, and these sporogenous tissues act as a pollen mother cells after few mitotic cell division, and this pollen mother cells undergoes meiosis, and it will form four pollen, which is known as a pollen tetrad, and at maturity each pollen are separated, is known as a pollen grain. So structure of pollen grain, we know that pollen grains represent the male gametophyte because it is the product of meiosis. That's why only n number of chromosomes. So it represents the male gametophyte. So it has two layers. The outer is known as the exine, which is made up of propollenin, the hardest biochemical substance, and inner one is known as the intine and made up of cellulose and packaging. So on maturity, the pollen gets contain two cells, the vegetative cells, the bigger one, and generative cell, generative very small one. At first it is attached in the pollen wall. Later on it is separated from pollen wall. So in case of 60% angiosperm, pollen grains are released from the microsporogenesis, these two cell space. Here we can see in left side diagram, this is the single pollen grain and asymmetrical division takes place. The larger one is vegetative cell, smaller one is generative cell, and after that when it pollinated, after pollination it germinated like this. So in the right diagram you can see here, so it is actually pollen mother cell, so it undergoes meiosis and it will form pollen tether, and this after that maturity it is separated, and this is a single pollen grain, and this single pollen grain will divide asymmetrically. So this is the vegetative cell and this is the generative cell. So here also in flow chart it is very clear, microgametogenesis. So what microgametogenesis is nothing but formation of male gametes from pollen grain. Already we have discussed that pollen grains it divide asymmetrically. The larger one is known as a vegetative cell and smaller one is known as a generative cell. After germination, this generative cell at the pollen tube it mitotically divide and it will form two male gametes. The vegetative cell generally form the pollen tube. In previous picture it's very clear. Here the vegetative cells after pollination it will germinate through germ pore, and this generative cell move forward to the pollen tube, and then later on it mitotically divide into two male gametes. So thank you for listening.